Welcome students. Today I will explain you how to analyze and measure biodiversity and how to do the various sampling of biodiversity and the various species all over the world. First of all, we need to start to understand what is a community. We have some different possibilities. One of them was proposed by Krebs in 1985. He said that a community is a group of population in a single place. Later, Began and other authors in 1986 proposed that the communities is just an assemblage of population which occur together in space and time. Now we need to summarize this definition, and we can express just the community as a group of interacting population in a single time in a single definite place. First of all, species in a community needs to interact with each other. This can include all species. So I mean the community can include all species or can be limited to a single guild. More common and more traceable, of course, because we reduce the number of species included. And a community can also be defined by a consistent special boundary. So, definition of community depends on how we design the studies and the sampling aimed as we want to understand and we want to calculate. There are different aspects of biodiversity. Before starting any kind of research, we need to ask ourselves, what can we measure? There are different possibilities, like we can measure richness of species, abundance of species, diversity, which is a combination or relationship between richness and abundance, the guild, evolutionary diversity with each species diversity, such as genetic, morphological diversity and other features. Then we need to ask how to summarize and describe the nature of this diversity, because we must get near about infinite number of things to record. So we need to simplify. Now the simplification is dictated by the experimental question, the location, the taxon, and the sample or the subsample from the nature. It depends on what we choose as an aspect of biodiversity, what kind of location we choose, or what life stage we choose. The sampling effort is just the number of samples we need to collect to ensure a correct sampling of the community. It depends on the economic resources which are available, the human resources, time, and the question to answer. It has been suggested like a sample of 10 is a rule. So it means that collecting 10 samples or plots or grades, etc., for each treatment would be enough or sufficient to provide a very good research study. 3, of course, is the magic minimal number in statistics, but is usually a too low sample number for ecology. So how to sample the data? We need first to keep data separately, because merging the data cannot be separated later. That's very important point. If you look for viability, you need statistics. That's the second very important point. But for doing statistics, if you merge raw data, you cannot perform any kind of statistic. So, for instance, if your sample bills per day, you need to take information per hour. Otherwise, later you can have this information but only together with day by day. To sample diversity, you need to form the population parameter. You need to remember that less sample you get, more biases you will have. So biases is just systematically different from the population parameter of interest. So it's important to get or to increase the number of samples to approach the real number of species. Now, if you are evaluating treatments, you need less samples. But if you are evaluating the total diversity, you need more samples. So, how to compare communities based on samples? You need to use abundance. That's the basic information for each taxon. For instance, if you are collecting species, genes, family, etc. All for any operational taxonomic unit. Then you need to show this biodiversity data. And there are different ways to show, which I will discuss in the later slides, because when I will talk about the sampling methods, I will mention it. But I want to introduce which system we have to explain to show biodiversity data. First is the rarefaction and accumulation curves. Now these curves just compare different samples with each other, and they make or give information about or how different are these samples from each other, and which one is the richest, which one is the poorest, etc. Another way is the log normal distribution of data. We can have different distribution of our abundance per species, and these can be shifted on the left. It means that we are missing some of the rare species. They also can be balanced, so they can follow a log normal curve. It means that we have a good representation of common as well as rare species. Another way to show biodiversity data is the rank abundance plot. The rank abundance plot is a plot that only shows the abundance rank of species, and on the ordinate the proportional abundance of species. And this is another way to compare different samples. A very useful tool is the species area curves. This curve, as it represents the area sampled, which is the on the x-axis, and the y-axis represent the number of species. So as soon as we get a kind of plateau, we can understand our sampling effort, or whether the minimal sampling effort is completed or not. So let's continue discussing how to sample biological diversity. Now, of course, it is impossible to collect all species in a community. So our aim should be to collect the greatest number possible of individuals, species, etc. A general rule is that it's better to collect many small samples than a few big samples. Another important thing is to ensure to get right and permission to study the sample. For instance, you have to ask national park or a protected area authorities to be allowed to enter the park and to collect your data for scientific purposes. You need to know statistics. That's very important point. And otherwise, if you don't know, okay, it's better you know something. It's better to learn how to carry statistics, but at least it's important you know or you have a friend who is a statistician. So in order to sample biological diversity, we need to select the target community. And for doing this, we need to choose the sampling frame. The sampling frame is just the range of the sampling that can be a political or geological or an administrative boundary. And we always need to specify in our research paper or report the sampling frame. When sampling biological diversity, it is very important to take care of the accuracy. Accuracy is the function of the bias and the sampling error. Also, of course, we need to mind the bias. We have two different types of bias: measurement bias and sampling bias. To get the number of samples, or if you vary the sampling technique, for instance, you have to increase the number of the species you are studying. So we need to keep in mind the sampling error. Sampling error can be detected unlike unlike the sampling bias. So you can detect sampling error. Measured and quantified and reported into the paper. How? How can you report a sampling error in your paper? By the statistical method of standard deviation or error standard. Sampling error is a kind of, you can say it is equivalent to genetic drift in evolution. Now, I have explained to you, when we are starting to sample population, then we draw and then we stop and draw and stop again, and we have the ratio that is changing from the beginning. Now then we need to select the sampling unit. A sampling unit is a square plot, a circle plot, a plot of grid, or something else. Choosing between design is a key question in biological diversity sampling. So how to choose between the sampling areas? It depends on the experimental question. For instance, if we are working in gradients, probably the best is to use the transects. Or in this case, you have to ensure the compatibility of different sample techniques. Now, if you are working in relative uniform sampling area, you would probably need to random your sampling. So, if done frequently enough, you need to get equal representation of the area that is included. So in that case, we may use the grid, which are very useful when needed for uniform samples in the area. Now, grid can be a good choice when we need to use regular intervals along two-dimensional design. Transect are very useful instead when we have to sample with reference to a straight line. Random sampling can be used to cite point quadrants or other sampling methods. There are other types of stratified sampling. For instance, as a very important element of the population, if you want a very important element of the population must be presented in only one stratified layer. Now we have different siting techniques. In short, a siting technique is just any viable form of collecting your samples. So we need to be sited at the level appropriate to the question. For example, you can use the point quadrate in the proximity to a central point within a cross. Now we need to use product if just sampling within a small area. We can use pitfall traps, beetle sheets, or even some kind of biological sensors. One of the most used sampling technique in plant survey are plots. Plots can be of different types. They can be nested, can be coarse plots, a Whitaker's plot. It depends on the sampling that we are going to organize. In animal survey, for instance, it's very important to understand how to work out the distance sampling technique. In this case, we just note the distance from our object, from the angle we spotted it, and the angle from our line, that is, for instance, where the animal was spotted. Now that is very useful, this distance sampling, because we can construct a kind of histogram in this way, and using this histogram we can estimate the number of animals that are present in the area. Another key aspect of biological diversity sampling is replication. Why we need to replicate? We need to replicate because we need to control for random and stochastic error. For instance, if I'm testing the independent factor that may otherwise determine the outcome of the experiment, then I need to increase the precision of my test. Now we need to increase the generalization of the test. So if you test across many sites, you can safely generalize to many others also. Now we have different definition of replicates. For instance, to consider a sample a true replicate, we need to maximize them in our experimental design. The greatest number possible, of course, and but with logistical limitation, we if you are very professional in what you are doing, then you need to use a power analysis. Otherwise, the subsample cannot can become absurd for the replicate. It means that this is only true if the subsamples are incorrectly treated as true replicates for statistical analysis. So subsamples are useful to increase the accuracy of the data estimate for the replicate. A special type of statistical analysis is therefore possible. So replication is due to I would like to say that if there is incorrect replication, for instance, when we are replicating samples and only samples but not the treatments, or when we replicate without independence. So the problem is that this violates the key assumption of statistical analysis. So for instance, the independent replicates, we need to increase the precision of studies if independent. We need to approximate through if vital if they are independent. We need to account for the normal random error. We need to allow set our key factor if it is suppose the alpha factor and keep it constant, and all of this are violated if our samples are replicated. So I will provide you an example. For instance, we have two treatments. The question is, what is the effect of treatment A and treatment B? Now if we do the replication, we treat each stock inside the square or your plot area That's our sample of the same color as a replicate. Now, if we do the right replication, we include only a single star of each color or their average. Now, it's very interesting to notice that almost 48% according to the right replication, and 71% of studies that are using ANOVA for to get rid of replication error. Now, that's a common statistical test and design for designing biodiversity experiments to get rid of errors. So this is important because particularly in this kind of studies, where we can have logistic problems, for instance, for certain animals, for transportation or foreign transportation problems might arise. There might be difficulty in calculation of certain animals which are not visible, or which are not in your range. This kind of problem may arise. But most of the studies are already in print. Some examples of replication when we have many samples from a single site are actually subsamples. That only a single for each treatment condition. Now these are actually replicas, but we cannot use statistics on a sample size of one or single sample from a single site. But replicating time would be true samples if the experimental question is time dependent. Vice versa, it just the replication. So how to control the replication? We need to know our question that time, whether the design includes replication. If we are working on taxonomy levels, it will be different from when we are working on the ecological level. So we clearly need to define our independent and dependent variables. What constitutes, for instance, if we are working on plant in a population, so we identify what is the true unit of repetition. Is very important for us to know the level of replication that is needed for the ecological studies. So we need to randomize our sampling design. This kind of randomization allows us to decrease sampling error and to increase the accuracy of estimation. There is a kind of stoppage rule on how many samples we should take for a good study. Someone suggested that we need to stop at the reference point that we can get from literature. For instance, some others suggested that we need to stop at the sample gathered from a statistical distribution of a ranked abundance plot. Another option is to select the minimal sampling area from the species area curve, or to estimate the sample number from the Alpha Fisher formula or other indices. So I hope this information which I provided you on how to analyze and measure biodiversity will help you to fulfill the tests that will be included later.